So meiosis is another form of cell division, and it is specifically only going to make eggs or sperm. So eggs and sperm are gametes that are going to be used in sexual reproduction. So this is going to be how we transmit genes from one generation to the next. So this is the beginning of heredity. How do we get offspring that look more closely related to their parents than they do to other unrelated individuals? And if you've looked at yourself and your parents, you'll probably note that you do indeed look like them. However, you don't look exactly like them. So one of the key important things about transmitting genes from one generation to the next is mixing them up a little bit. You want to pass your genes on, but you want them to have a little bit of variation so that the population is widely varied. So not only is meiosis going to allow us to transmit genes from one generation to the next, it's also going to allow us to create genetic variation, which is extremely important in maintaining the variation within a population. So genetics is the study of heredity and variation, which we will get into in the next unit. So a gene is just a segment of DNA. It usually is going to code for some specific protein, and these are units that are heritable. So we're going to pass our specific genes onto our offspring. However, we may not give them all of our genes. We give them some of our genes, and we might give them some genes from the other parent. And so these are passed through reproductive cells called gametes. So these gametes are sperm and eggs, and our DNA is packaged into linear chromosomes. So again, humans have 46 chromosomes. A sperm cell and an egg cell are haploid, meaning that they're only going to have 23 chromosomes. That way, when you have a sperm and an egg, which each have 23, and they come together to form the next offspring, that's going to have 23 plus 23, which is going to give us our 46 chromosomes that we need to have for body cells. So meiosis is going to be involved in sexual reproduction. Sexual reproduction is going to involve two parents, and it's going to involve a unique combination of genes. So each parent is going to produce gametes, eggs, or sperm, and those are going to be mixed up in some way to create a unique combination. So unless you have an identical twin who looks exactly like you, if you have a sibling, they look probably slightly different. They might have different eye color, hair color, height, or just different characteristics. And that's because every time parents create eggs and sperm, they get a slightly different combination of genes than the previous offspring. Asexual reproduction is what we already covered in mitosis. So this is a single individual bacteria reproducing by just making copies of itself, or a single body cell creating more body cells. So these are clones. So this was chapter 12. When we look at a set of human chromosomes, and you can see this over here, is called a karyotype, and we do this when we do genetic testing, especially on infants, to see if they're going to have any chromosomal disorders. So you can see that each chromosome is numbered, and most human chromosomes are numbered based on size, and they are paired. So chromosome 1 and chromosome 1. The reason you have 2 of each chromosome is you got one from mom and you got one from dad. So for each of these pairs, you got one of those chromosomes from your mother and you got one of those chromosomes from your father. These sex chromosomes are over here. So we have 22 pairs of what are called autosomes. So those are the body chromosomes, so they're numbers 1 through 22. And then we have one pair of sex chromosomes, and those are the sex-determining chromosomes. In this case, X and Y would create a genetic male, an X and an X would create a genetic female. These pairs are called a homologous pair. So in meiosis, it's going to be important to make sure that the egg or the sperm only gets one copy of each chromosome. We don't want to just have a grab bag where we grab 23 chromosomes randomly and give them 2 copies of chromosome 8 and no copies of chromosome 11. So in meiosis, what we're going to do is we're actually going to get these homologous pairs of chromosomes to find each other, and then we're going to split them up from their pair so that we know that each cell is going to get chromosome 1 through 22 and then an X and a Y. So the sex chromosomes determine the genetic sex of the individual. Typically, human females are going to have an X and an X. Typically, human males are going to have an X and a Y. The Y is actually going to determine maleness, so Y has genes on it that trigger the production of male genitalia and male hormones. Again, the remaining 22 pairs are called autosomes. Auto means self. Soma means body. So those are usually going to be associated with all of the different body characteristics that aren't typically related to sex. The X chromosome does have genes on it that are related to things like color blindness and other body chromosomes, as the X chromosome is much larger than the Y chromosome. So again, for each pair, we have one chromosome from each parent. So in the X, let's say we have an X and a Y, this is a homologous pair. We got one of them from mom and one of them from dad. So here we see these cells, diploid and haploid again. A diploid cell has 2 sets of chromosomes. So these are going to be the somatic cells. The somatic cells are a precursor to eggs or sperm. So you have a diploid pre-egg cell and a diploid pre-sperm cell that has 2N that's going to have to go through meiosis to eventually become eggs or sperm. So a somatic human cell is going to have 46 chromosomes. A haploid cell, again, this is going to be our gametes. So this is going to be the eggs or the sperm that we make at the very end of the meiosis process. For a human, these gametes are going to have 23 chromosomes. So from a description of a cell, you should be able to determine if it is haploid or diploid based on how many pairs of chromosomes it has and information about the cell. So if I tell you a cell has an N of 3, that means that it normally, as a diploid cell, 2N would have 6. And as a haploid cell, 1N would have 3 chromosomes. So that's what we're going to use here for our example of just walking through the steps of meiosis. And you can see it has color-coded here. We'll just number these chromosomes 1, chromosome 2, chromosome 3, chromosome 1, chromosome 2, chromosome 3 by size. So it has 2 copies of chromosome 1, one of those from mom, one of them from dad. It has 2 copies here of chromosome 2, and then 2 copies here of chromosome 3. And you can tell that these have been duplicated in preparation for dividing. So this cell here is diploid because it has 2 sets of chromosome. It's got one set of 1, 2, 3, and another set of 1, 2, 3. So 2N equals 6. So let's talk a little bit about the reproductive life cycles. So hopefully you're familiar with the human life cycle and how that happens. So what happens first is we're going to go through meiosis, and we're going to form eggs and sperm. These are our gametes. In the process of forming the next generation, so if you were going to have a baby, you would go through fertilization. So the very first step of fertilization is eggs and sperm's union coming together to form a single cell called a zygote. And the zygote has one set of chromosomes from each parent, which means it has 23 from mom and 23 from dad, which is a total of 46 chromosomes. So is a zygote haploid or diploid? A zygote is going to be a diploid cell. The zygote is then going to produce more cells through mitosis. So the zygote goes through mitosis to form eventually an infant, which is going to be birthed and eventually grow up into a mature adult. Once this infant has grown up into a mature adult, it's going to go through puberty, and at puberty, it's going to go ahead and start making gametes through meiosis, whether it's producing an egg each month or producing sperm cells constantly. Those gametes can then combine with the gametes of a different individual through fertilization and then continue the life cycle. Meiosis is extremely important for maintaining the chromosome number. Meiosis cuts the chromosome number in half. So when we take this adult and we go through meiosis, they're going to go from having 46 chromosomes in a body cell to having only 23 chromosomes in an egg or a sperm cell. And that's important. If we don't cut the number in half, then their offspring would have too many chromosomes. If we just left it at 46, then we would double the number of chromosomes in every generation, and that doesn't work well. You wouldn't have a baby. And so meiosis is important for maintaining the fact that humans have 46 chromosomes. If we don't cut them in half, we don't maintain that number. There are other sexual life cycles, so you're probably most familiar with the human life cycle, which is the animal life cycle, which is what we just went through, where we have gametes, so we might have an egg and a sperm from two different individuals come together and form fertilization. We then create this diploid individual. So you can look at the size of this to see how many cells there are. A really small cell is going to be unicellular, right? So a sperm is one cell, an egg is one cell. And then a larger cell here is going to be multicellular. So we have a multicellular diploid phase, which is our adult body. And then eventually we go through meiosis to create more eggs or sperm. Plants and algae go through a different type of life cycle called the alternation of generations, where they actually have two different multicellular stages. The big part of like a tree or grass or plants that you're seeing outside are usually this large diploid body, which is called the sporophyte, which creates haploid spores. And then they actually have a haploid body called a gametophyte, which is then going to create haploid gametes. So this would be the eggs or the sperm. You'll go into this alternation in generations in a lot more detail in biology 2, but as far as biology 1, what you really need to know is that animals only have a multicellular diploid stage, whereas plants have a multicellular haploid and diploid stage. So they have two multicellular bodies. The opposite of the animal life cycle is the fungus and then some protists. So think of like mold, bread mold would be a fungus here. They are zygotes for a very short amount of time, so they do not have a large diploid body. Most of what you see as mushrooms or mold are actually a large haploid body. So they have a multicellular haploid phase and a very, very brief single-celled diploid phase. So this is kind of the opposite of the animal life cycle. So now we're going to go through all of the steps of meiosis. And meiosis, just like mitosis, has replication of chromosomes. So in interphase, we're going to go through the S phase, which is going to copy the DNA into multiple copies. Unlike mitosis, meiosis is going to have two cell divisions. So this makes sense. We're going to have two copies of DNA, but we need to cut the number of DNA in half. So we're going to cut it in half once with one division called meiosis 1, and then we're going to cut it in half again with the second division called meiosis 2. So instead of ending up with two daughter cells that are exact clones like mitosis, we're going to do two divisions to end up with four daughter cells that have only half as many chromosomes. So we're going to start with one diploid cell, and we're going to end up with four haploid daughter cells, which will be our sperm or egg. So let's go through meiosis 1 first. So meiosis 1 is going to have prophase, metaphase, anaphase, and telophase 1, followed by cytokinesis to separate them into two cells. We're not worried about pro-metaphase at this point. You can take prophase and pro-metaphase and kind of collapse them just into prophase. So we've got P, M, A, T, prophase, metaphase, anaphase, and telophase. So up first is prophase 1, and this is going to So meiosis 1 is all about separating homologous chromosomes. So in the very beginning of meiosis, we have to find the sister chromosomes that belong to the same number of chromosomes. So if this is chromosome 5, this is chromosome 5. This is our homologous chromosomes. Those are going to find each other. So you can see here in prophase, we've got some other things that are very similar to prophase. We've got the centrosomes kind of migrating to the different poles. We've got the spindle fibers forming to separate those. We've got the nuclear envelope and nucleolus disappearing. But notice all of the chromosomes. The chromosomes have started to condense, and they are formed in their homologous chromosomes. So when the homologous chromosomes find each other, this forms what is called a tetrad, because it's got 1, 2, 3, 4 sister chromatids in a tetrad. Tetra means 4. And then I want you to look at what these chromosomes are doing. They're not just sitting right next to each other like my picture. They're actually forming little X's. They're crossing over each other at these little regions called chiasmata. And this is in preparation for something called crossing over. So crossing over is only going to happen during prophase 1. You absolutely need to know that. That'll definitely be on the test. And what happens is the chromosome from mom and the chromosome from dad find each other. And the chromosomes are going to cross over. And then a little portion of those is cut off, and they actually switch pieces. So the non-sister chromatids, those are the ones in the center next to each other. Non-sister means that we've got one from mom and one from dad. These are sister chromatids. Those are not exchanging because those are exact clones of each other. What we're doing here is we're creating some genetic diversity. We're going to switch chromosome pieces from mom and dad and make new pieces of chromosomes that are actually combinations of what we got from mom and dad. So this is called synapsis, which is where the DNA pieces actually break off, switch pieces with each other, and then reform. So if chromosome from mom is red and the chromosome from dad is blue, after crossing over, we're going to actually have new combinations of chromosomes where these two have switched places. And so now we have a sister chromatid that's purely from mom, a sister chromatid that's got mom and a piece of dad, a sister chromatid that's mostly dad with a piece of mom, and a sister chromatid that's completely from dad. So all 4 of these are going to create different versions of this chromosome in the ultimate daughter cells. So crossing over happens in tetrads in prophase 1. Next we have metaphase 1. So what's going to happen in metaphase 1 is that the homologous chromosomes are going to line up at the metaphase plate. Notice that we are still in those tetrads. The homologous chromosomes are still associated with each other. Anaphase 2, the homologous chromosomes are going to separate. So at this point, we're going to separate. We're going to pull one full chromosome this way and one full chromosome this way. And notice again that you can see these little pieces where they went through crossing over, where this chromosome and this chromosome have slight differences now because they shared some DNA in crossing over. The reason that we have to do this first process where we separate homologous chromosomes is so that the daughter cells are going to have one copy of chromosome 1, one copy of chromosome 2, and one copy of chromosome 3. This one has one copy of chromosome 1, one copy of chromosome 2, one copy of chromosome 3. So this is going to ensure that the 2 cells that we make here have not just 3 chromosomes, but the 3 correct chromosomes. So in telophase 2, we see very similar. The homologous chromosomes are now in a new... The homologous chromosomes have been separated, and they're now going to form new nuclei. Sometimes the nuclei fully reform here and then have to break down again. Sometimes they don't completely fully reform because the cell is going to divide again. It just depends on the type of cell. And then eventually we'll go through cytokinesis and cleavage furrow, which will give us 2 daughter cells. These cells are haploid at this point. So the cell in prophase 1 had 1, 2, 3, 4, 5, 6 chromosomes. So this has 2 pairs of n equals 3 chromosomes. So this is 2n equals 6. Each of these cells is going to only have 1 pair of chromosomes. So this has got 1, 2, 3 chromosomes, which is n equals 3. So by the end of meiosis 1, those cells are haploid. However, the DNA is still duplicated. So now we're going to need to divide again to break apart the duplicated DNA. Mitosis 2 is going to be very similar to... Meiosis 2 is going to be very similar to mitosis. So in meiosis 1, we had the separation of homologous chromosomes. Meiosis 2, we're going to have the separation of sister chromatids, which is just like what we did in mitosis. So again, the phases are named the same way, PMAT. So prophase 2, and this is happening twice because we already divided once. The chromosomes are condensing again. The nuclear envelope is breaking down. The spindle fibers are forming and moving to the opposite poles. This way we're going west and east this time. And this is happening in both cells at the same time. Metaphase 2, the sister chromatids are going to line up in the center. This should look just like it did in mitosis. So these cells start out with 3 chromosomes. These are haploid. This one is haploid, right? 3 chromosomes. Break apart here for a second. We have 6 chromosomes inside of a cell. So those cohesins that hold the centromeres together get broken, and the chromosomes are going to be pulled to the opposite sides. And then in telophase, each one of these cells is going to have 3 chromosomes. So meiosis 2, it starts haploid, it ends haploid. We actually go from diploid to haploid in meiosis 1. So again, here the nucleus is going to reform, our spindle fibers disappear, we're going to go through cytokinesis, and then we will end up with 1, 2, 3, 4 haploid daughter cells between our asexual reproduction and our sexual reproduction. So mitosis conserves the number of chromosomes. This is going to make clones. Whereas meiosis cuts the number of chromosomes in half. We go from 2n to n, haploid to diploid to haploid. Mitosis are going to have genetically identical to the parent. Again, these are clones. The goal of meiosis is to create genetically different individuals so that we can preserve variety in the population. In mitosis, if you've got a skin cell and you're just trying to repair skin cells, you definitely don't want to make different skin cells. You want to keep them exactly the same. Mitosis, we have the cohesins cleaved apart during metaphase. This means that we're separating sister chromatids. Meiosis, anaphase 1 separates homologs, those tetrads, whereas meiosis 2 is going to separate sister chromatids. This is something that you absolutely should know. This will definitely be on the test as to whether you're separating homologous chromosomes, those pairs of 4, or just sister chromatids. Mitosis has 1 division that's going to result in 2 cells, 2 daughter cells. Meiosis has 2 divisions that's going to result in 4 daughter cells. Meiosis really has 2 goals. The first goal is to cut the number of chromosomes in half so that each generation has the correct number of chromosomes. The second goal is to create diversity. So genetic diversity is really, really important in populations. So let's think about the coronavirus pandemic. If every single human on the planet had the exact same genetics and was equally susceptible to COVID, it could be even worse. Every person was really susceptible, then we would end up with everybody dying of COVID. But that's not the case. Humans on the planet are extremely diverse. Even within a single country, individuals are very, very different. And that's because of genetic variation. So genetic variation results in some people who are very susceptible and some people who are not. And that is good for the overall population. We will survive the COVID pandemic and move on. Genetic variation is really, really important for populations to make sure that those populations persist. So that is the second main goal of meiosis. We don't do this in mitosis because mitosis is not new individuals. Changes that happen in mitosis are usually called mutations. So mutations are changing the actual DNA, which creates different versions of those alleles. Sometimes those are good, sometimes those are detrimental. That's not what we're talking about with meiosis. With meiosis, we're not changing the genes, we're just reshuffling them. So instead of getting the genes that mom had, maybe you're getting a portion of mom's genes and a portion of dad's genes, and so you have a different mixture than what each parent had. There are 3 places where genetic variation can occur in meiosis. I often ask this as an essay. So the first is independent assortment of chromosomes. The second is crossing over, and the third is random fertilization. So all 3 of these mechanisms are going to create genetic variation, not through mutation, but through reshuffling and reorganizing the genes from 2 parents into a new offspring. Independent assortment of chromosomes is talking about how the homologous chromosomes line up at metaphase, and this is randomly. So all I mean here is which chromosome is on the left and which chromosome is on the right. Is this one from mom and this one from dad? Or is this one from dad and this one from mom? What's going to happen is there's equal chance of this scenario or this scenario. And so for every number of homologous chromosomes, there are 2 ways that it could line up. Mom on the left, dad on the right, dad on the left, mom on the right. Humans have 23 pairs. So we actually have 2 to the 23 number possible combinations for our chromosomes to organize themselves. And this again happens randomly, which leads to 8 million different combinations of arrangements. So here's what this looks like. Here's a cell that only has 2 pairs of chromosomes. So this 2n is equal to 4, n is equal to 2. So this is a diploid cell. This would be during metaphase 1, where our homologous chromosomes are lining up. In this one, the chromosomes from dad, again, they're color coding, blue is dad, red is mom. The chromosomes from dad both lined up on the left, chromosomes from mom both lined up on the right. The opposite could be happening. We could have one of these switch, where the chromosomes from mom down here for chromosome 2 was on the left, whereas the chromosome for dad is on the right. So these are going to result in 2 different possibilities. These are 2 equally probable arrangements. So what's going to happen in metaphase 2 is we're going to have one of their daughter cells over here that has both chromosomes from dad, both chromosomes from mom, and this one, each daughter cell is going to have 1 chromosome from each parent. So we end up with daughter cells where we have the haploid chromosomes could be both from dad, both from mom, 1 dad, 1 mom, or the opposite, 1 mom, 1 dad. So there are 4, 2 to the 2, different combinations of gametes that can be formed depending on the way that these arrange themselves. And that's just with 2 pairs of homologous chromosomes. So we have 2 to the 23 of combinations of ways that our cells can arrange themselves to end up with different combinations of chromosomes in the daughter cells, and that's without mixing up any of the actual DNA. The second mechanism of crossing over is recombining chromosomes. So this is where we're going to actually take pieces of the chromosomes, they're going to physically cross over each other and switch. This is not rare. In humans, we get 1 to 3 crossover events per chromosome. So you have 23 chromosomes. This is going to happen 1 to 3 times per chromosome. So this is very, very common. So here we go. We have our tetrad here. We have non-sister chromatids that are next to each other. They are going to physically cross over. The synapsis is the process of them breaking off and switching. And so in the end, we have this chromosome is fully from dad. This one was originally from dad, but now it has 2 pieces from mom. This one was originally from mom, but now has 2 pieces from dad. And then this one is fully from mom. So we have 4 different chromosomes that are going to result from meiosis II. Add these genetic changes to the already 8 billion combinations of chromosome arrangements, and you can see that we are just magnifying greatly into the tens or hundreds of billions of possibilities for each egg and each sperm. The last variability is random fertilization. So this is which egg gets produced and which sperm is the one that's going to be able to fertilize it. We're going to produce lots of gametes. So women produce typically 1 egg each month or each menstrual cycle. However, we produce 4 cells, and then one of those randomly is going to develop into the egg, whereas the other 3 are randomly going to be reduced to polar bodies that aren't going to develop into an egg. So each month, even though we only produce 1 egg, it's still a random chance of which one of those 4 cells is going to actually become the egg. Males produce millions and millions of sperm. Each one of those sperm has a chance at being the one that actually fertilizes the egg. The fusion of 2 gametes, so each egg or each sperm has 8.4 million possible chromosome combinations from just independent assortment, and more from crossing over, is going to produce a zygote, right? So we have fertilization of egg plus sperm is going to make our 2N zygote. And we have about 70 trillion zygote combinations based on which egg and which sperm was the one that actually was able to fertilize. So this is why if you have a sibling, you probably still look slightly different from your sibling, because even though it's the same genetic pool of mom and dad, you have 70 trillion different ways to combine those pools to make new individuals. So unless you're an identical twin, you're going to have a different genetic makeup than a sibling. Genetic variation is extremely important because that's how natural selection works. Populations are not going to be able to evolve and adapt to environments unless we have a pool of genetic variations. The only way to make new genes is through mutation. However, sexual reproduction is going to maintain our genetic variation in populations. Most organisms that reproduce sexually or have some method of reproducing sexually. It's not common for animals to have only asexual reproduction. There is one here, this is a rotifers, that's an animal, and it actually only reproduces asexually. However, they do have mechanisms of incorporating foreign DNA from dead individuals in their environment into their body. So even though this is an animal that reproduces asexually, they always have some kind of mechanism of maintaining genetic variability. Same thing goes for like protists and bacteria that reproduce through things like binary fission, which is cloning. Those kinds of organisms have different mechanisms like conjugation for sharing DNA with other individuals to maintain genetic variation in the population.